Hello, everyone. Welcome to the last fan bug of this season. I'll be your host. My name is Jonathan. So, as a reminder to all our Zoom attendees, please mute your mics and phones if you're here in person. This event is being recorded, and you heard the announcement earlier. This will be today's schedule. We'll start off with announcements, then our introductory training talk, followed by our feature talk, and then we will have dinner and social. VanBug is primarily a volunteer-driven organization facilitated by 3 faculty members from UBC and SFU, Dr. William Chow, Dr. Amy Lee, and Dr. Faraz Hodge. Without them facilitating, we wouldn't be able to organize this series of seminars. So with the final seminar, also means the beginning of the next season, we will be announcing the dates on vanbug.org in late summer. And as we're still recruiting, this is your final chance to suggest a speaker for us to invite for next season's fanbug seminar. And you can do that by visiting fanbug.org and clicking the contact tab and clicking suggest a speaker. Likewise, in the same page, you can also subscribe to our mailing list to keep yourselves informed of all things VanBug. And you don't miss out by subscribing to our calendar as well as our social media. We will upload all of our recorded talks on YouTube. VanBug is sponsored by the Graduate Studies in Bioinformatics at UBC and SFU, Genome British Columbia, the Big Data Hub at SFU, the Michael Smith Laboratories at UBC, as well as Langara and Stem Cell Technologies, as well as Vancouver Prostate Center and VGH. VanBug is also affiliated with the ISCB and our sister bugs, MonBug and TorBug. And we're also the proud partners of the Canadian Bioinformatics Hub British Columbia node. They offer training, community, and career for people interested in bioinformatics, and these are QR codes if you're interested in looking up further resources. For our Zoom audience, please ask your questions in chat, and a VanBug volunteer will voice it for you. We will begin with a talk from Neera Petadia, who's a PhD candidate in bioinformatics, and her talk is titled Examining Differential Expression Patterns in a Large Data Corpus, and followed by our feature talk from Dr. David C. Clarke. He's an associate professor at SFU, and his talk is titled Towards Manipulating How Muscles Grow, Bioinformatic Inference of Exercise Response Control of RPS6KB1 expression.
Sorry. Yeah, so hi everyone, my name is Nira, and I'm going to be talking a little bit today about my project looking at differential expression patterns in a large data corpus. So as I was preparing for this presentation, out of curiosity, I looked up how many human data sets were in GEO. And if you look up here at the number in the top, you can see there are quite a lot. And over the years, the scientific community has published thousands of expression studies and amassed a very vast collection of transcriptomic data. We trust that the individual results from these studies provide some type of genuine biological insight into whatever condition is being looked at. And I expect that because we can identify biological signals from individual studies, we should be able to identify signals when we look across many studies. However, figuring out exactly how we go about doing that and interpreting all this data as a whole remains to be a pretty significant challenge to the field. And this is something that I try to address with my project and we'll be talking about today. So one of the things that does make it easier, to go about addressing this problem is to work with good, clean data, like using a resource like GEMMA. So GEMMA is a curated resource of transcriptomic data that we have been working on in the Pavlidis lab for the last 20 years or so. And so we basically pull data sets from GEO and the SRA. We go through reprocessing and annotating them using standard pipelines to basically allow users to pull this data and look at it, do whatever analyses they're interested in. So the data sets that we have in GEMMA cover a very wide range of different biological topics. And as of 2025, it contains over 20,000 experiments. So now that we've spent all this time collecting and cleaning all of this data, how can we go about using it? So to kind of demonstrate this idea, suppose that this is a result from my experiment where we can see what genes are being differentially expressed and what genes are not being differentially expressed. Just looking at this experiment alone can be difficult to actually go about trying to interpret what exactly is going on within the condition. But what we can do is we can compare the differential expression results from this one condition across many conditions. And what we can see here is, for instance, gene A, while it is differentially expressed in our condition, it's also being differentially expressed in a whole bunch of other conditions, in contrast to gene H, which is only being differentially expressed within just a single condition. So this sort of leads me to my two main questions I want to talk about today. What genes are commonly differentially expressed across all conditions, and are commonly differentially expressed genes involved in specific biological processes? So just out of curiosity to the room here today, how many of you guys have gotten one of these genes or a gene on this list as a result in your differential expression analysis? And you can raise your hand. Okay, see quite a few hands that are being raised. So where did I get this gene list? How did I know that the genes from your experiment would be on my list? So these genes come from something called the DE prior, which was introduced in a publication by Crow et al. This was done in collaboration with our lab group. And essentially what was done in the study was we ranked how often genes were differentially expressed across many experiments, looking at different biological conditions, and found that there were genes that showed frequent differential expression regardless of the biological question being addressed. This ranking was then used to interpret a variety of different things, such as specificity of different biomarkers, interpreting known gene sets to examine how specific those genes were to whatever disease that was looking at in that gene set, and also the interpretation of differential expression results from meta-analyses. Now, to compute the DE prior, we basically score each gene based on its frequency of appearance in differential expression results, or hit lists, while weighting the frequency by the size of the specific differential result in which that gene appeared. Once we've computed these scores, we then convert them into a ranking where genes that are ranked close to 1 can be thought of as being commonly differentially expressed. So since the original DE prior publication, the GEMMA has changed quite a bit. We have nearly 4 times the amount of data that was available back in 2019, and we have drastically increased the coverage of data sets from other platforms, particularly RNA-seq. Additionally, the original DE prior was only computed using the human data that we had within GEMMA. So in my project, one of the things that I wanted to do was recompute the DE prior leveraging more data, and also being able to compute the prior for both human and mouse. So the first thing that I wanted to do was to see which genes ranked highly in the DE prior for human and mouse. So what we can see here on this plot, each point here represents a one-to-one orthologous gene pair between human and mouse. And the x-axis is the prior ranking for the human genes, and the y-axis is the prior ranking for mouse genes. We can see that the human and mouse priors are highly correlated with each other. And now if we go ahead and look at some of the top-ranked genes in the prior, if we go ahead and look at some of the top-ranked genes in the prior, we can see that there are certain themes of genes that pop out, such as sex marker genes, immediate early genes, stress response, and inflammatory response. So these types of genes are commonly being differentially expressed across experiments that look at a variety of different biological topics. So then the next thing that I was interested in looking at was trying to see which genes had a high DE prior ranking in either human and mouse. So here, what I have plotted on the right is the difference in the prior ranking between human and mouse on the x-axis, and then the difference in proportion of differential expression results that had the gene as being differentially expressed, again, between human and mouse. So basically on the bottom quadrant, the genes were ranked higher in mouse, and in the top quadrant, they were ranked higher in human. Now, this is something that we are interested in looking in further, trying to see, what if there are particular conditions that maybe drive why certain genes may be more frequently differentially expressed between mouse and human, if it's, sort of a data-related fact, and also seeing if there's any commonalities between these different gene sets between human and mouse. So then the next question that I wanted to look at was, is the DE prior ranking biased by unequal representation of conditions in GEMMA? So in GEMMA, we don't have equal representation of every condition. So for instance, if we take a condition such as dementia, there are 200 differential expression results versus for other conditions, there are far fewer, maybe like 10 or 15. And we can also see just from this plot here that the variance is quite large in the actual representation of these conditions. So the question that I had was, are high DE prior genes differentially expressed specifically in the overrepresentation of the conditions? And that's why they tend to rank higher within the prior when we compute it. So what I did was for every single gene, I compared the condition-specific differential expression for the gene and compared it to its global differential expression frequency across all of the differential expression results that we have within GEMMA. And I've also highlighted the genes that rank in the top 1% of the DE prior. And what we can see in a variety of these top high-ranking conditions, the top DE prior genes that are showing up are showing up both as being highly differentially expressed in the specific condition and also are showing up as being highly differentially expressed globally as well. So what that means is that the DE prior is not necessarily being driven by one specific condition or the overrepresentation of one specific condition. Or is it? So there is a notable counterexample to this. Biological sex, so comparisons between male and female, have certain genes that are specifically differentially expressed or differentially expressed at high frequency within or across a lot of the contrasts that are male versus female, but not really differentially expressed in a lot of other studies. And this is one of the largest categories that we have within GEMMA. And this in a way makes sense because these genes have very, very specific differential expression. And also, there are a lot of studies where people do comparisons between, do biological sex comparisons. So just to wrap up, some key takeaways. In my project, I have recomputed the DE prior, leveraging much more data from the original Crow et al. Prior, and we found that the human and mouse DE prior rankings were highly correlated with each other. We also identified some genes that appear to be species-specific in terms of their prior ranking. And generally, the DE prior is not driven by the overrepresentation of a specific condition, but biological sex is a notable exception to this trend. So in terms of next steps, there's a couple things that I'm interested in doing. One of them is to see what's going on in the middle of the DE prior. So these are genes that are maybe have a ranking that's, in the middle section. And I'm curious to see if those genes tend to be differentially expressed between a specific biological condition, or if that might just be some level of noise within the data. I also am curious in seeing what the effect is in regressing out specific conditions from the DE prior and how that affects the ranking. So for instance, if we removed all the sex comparison differential expression results in the prior, would the sex genes still show up at the top of the ranking? And finally, because in GEMMA, we've started the process of being able to add single cell data into our database, I'm curious to do parallel analyses in single cell and see if similar themes of genes also show up at the top of the ranking. So that brings me to the end of my talk. I just want to thank all the help from my supervisor, Dr. Paul Pavlidis, and also all of my lab members, particularly Nathaniel Lim, who is a former PhD student in our lab who really laid the groundwork for a lot of the stuff that I'm doing in my project. So yeah, thank you. Yes.
Human and mouse different lifestyles. You see that age.
Yeah, so just like in terms of looking at like if there are more comparisons of age between human and mouse. So that is something that we can definitely look at. However, I don't expect it to play a massive role in the ranking of the prior. Mainly just because when we look at, the trends that we're seeing in the data overall, we see this sort of consistent pattern of how these DE prior genes rank. But, I think it's an important thing to consider, like this is something that's entirely based on the data. So, it's important to understand like what the effects of the actual conditions themselves and other, biological artifacts such as age differences and what that role that's playing in the data. Yeah, exactly. Yeah. I think like the hope with that is that because the data that we're working with is so large and so heterogeneous that, some of these like more nuanced differences are things that, will get kind of washed out with like the volume of data that we have. I'm so sorry, I can't really hear what you're saying.
So if someone carries out a differential expression analysis experiment, they give less weight to that finding because
Yeah, so I think in this work, I'm not necessarily proposing that people like throw out certain genes that they're looking at. What I'm really trying to say is that interpreting our differential expression results are very challenging, and this is just another way we can go about trying to understand, why certain genes are showing up as being differentially expressed in our studies, and it can help contextualize why we're getting the results that we're getting.
Okay, thank you.
Hi.
Hi.
My question is, so what is a condition? Like, how do you define it? Like, is every single thing that you can think of a condition, or like, do you have any criteria, or what you consider a condition?
Yeah, of course. So I'll just kind of go back. I do have a bonus slide. Goes back to my first slide. So a condition in GEMMA, what we're referring to is like the very specific topic that we're looking at, like Alzheimer's disease, breast cancer, Parkinson's, whatever it is. It's not just limited to diseases. It could be specific drug treatments. How we typically define a condition is based on the annotations that we're getting from the data in GEO. So like what's in the metadata from it. More often than not, the what's being tested in the study is usually within the metadata. It's usually reflected some kind of like formalized ontology term. And so that's what we're representing as the condition. Another way you can think about it is, what's the manipulation that's being done in the study? So, is it like Alzheimer's versus control patients? Is it drug one versus versus no treatment? So yeah, does that help?
Yeah. Yeah, thank you.
Thank you. Thanks for listening.
Yeah. And now we have our feature speaker. Yeah. I'll introduce you. Dr. Dave Clarke is an associate professor in the Department of Biomedical Physiology and Kinesiology at SFU. Dr. Clarke holds a bachelor's and master's degree in kinesiology and a PhD in chemical and biological engineering. He directs the laboratory for quantitative exercise biology, the mission of which is to build predictive models of exercise training adaptations to improve fundamental understanding and to optimize exercise training programs for health, fitness, rehabilitation, and or performance goals. To achieve this mission, the lab pursues 3 primary lines of inquiry: exercise responsive cell signaling networks, training load quantification from wearable technology data, and evidence-based exercise programming.
Okay, thanks very much for having me and for showing up tonight on a nice spring evening, and thanks, Jonathan, for hosting. So yeah, so I'm going to speak today. So just to set the stage a little bit, I'm very much a bioinformatics user, not so much a developer. So we're kind of, you can ask me about some of the details of the methodologies, but they may be a little thin in responding, but we'll do our best. Just before I get going, I just want to acknowledge that this event is taking place on the traditional lands of the Squamish, Tsleil-Waututh, and Musqueam First Nations. So some of our efforts towards reconciliation in our program include indigenizing our courses, and also, for example, like the occupational physiology of subsistence living, as well as some of the cultural and recreational practices. We're also trying to foster the integration of kinesiologists and exercise professionals in communities to help improve the health and rehabilitation of community members, and also training Indigenous persons as exercise and kinesiology professionals to serve their communities. Okay, so what I'm going to talk to you today is about a protein, well, the gene that codes for a protein called P70S6 kinase. It's a protein that governs the translational initiation and efficiency, which is important for skeletal muscle protein synthesis. We've done some computer modeling studies of muscle protein synthesis, and these studies predict that the absolute levels of this protein are critically important for controlling the rate of muscle protein synthesis. And then to follow that result up, we applied bioinformatic analysis of the gene to better understand its transcriptional control. So the talk is divided into 3 parts. I'll introduce P70S6 kinase and how it's a controller of muscle protein synthesis. I'll then go into the bioinformatics programs that we use, or the algorithms, centering on something called Carnival. And then we'll summarize some of the insights that we derived for exercise responsive signaling and S6 kinase gene transcription. All right, so I'm not sure to what extent you guys are interested in skeletal muscle, but I certainly would advocate that you develop an interest in skeletal muscle because it's a really fascinating tissue, and it's responsible for our survival, optimal health, and quality of life. So it's the tissue that allows for locomotion. It therefore enables physical performance. It's also extremely important for our metabolic health. So by virtue of its mass, skeletal muscle is the largest organ in the body, and it's a massive reservoir for blood glucose. So, the reason that we can have blood glucose homeostasis is in large part because we have skeletal muscle cells that take it up. Okay, and so when there's also a reservoir for amino acids. So under conditions of starvation or metabolic stress, it will contribute amino acids for gluconeogenesis in keeping the brain well fed. It's also going to help produce heat. And then also it's an endocrine organ. Muscle secretes all sorts of hormone and paracrine-like substances, even in response to exercise. And in fact, a lot of the health promoting effects of exercise on organ systems outside of the heart and skeletal muscle and other tissues that are directly involved are due to these endocrine-like effects. And lastly, something you may not have considered much, but in the agricultural industry, muscle is of extreme interest because it's the basis of meat. Okay, and nowadays, of course, we have the engineering of meat and culturing of it in a dish. And so understanding muscle's properties and in particular its development ex utero, like sort of ex vivo, is of significant interest. So what is skeletal muscle? This is kind of the anatomy. So if you look at sort of a whole, this is at the whole organ level on the one side, and maybe I'll use the pointer instead of standing here, so for the people on Zoom. But you can see how it's kind of a very like hierarchical type of tissue. And its major constituent is the myofibrils. And these are the contractile proteins, so actin and myosin, the filaments that slide past one another during contraction. Outside of water, protein is the largest constituent of muscle, and most of that protein is the contractile proteins. So the way that muscle mass is directly a function of skeletal muscle protein. And the way that muscle protein levels are set is basically the emergence or the emergent levels from the difference in the rates of synthesis and the rates of breakdown. And both are happening all the time. Okay, so your muscles are constantly turning over. Now, when you feed, you eat as part of that protein, it gets digested, broken down into amino acids. The amino acids go to the muscle. They're taken up, and then they're incorporated into the protein. Similarly, the protein's broken down into amino acids. But with feeding, the rate of synthesis is going to be higher than the rate of breakdown. So when you eat a meal, you're building up your muscle, in the hours after. But then during that postprandial period, when you're kind of in the fasted state, you're actually losing muscle mass at that point. And it's this daily interplay of like eating 3 times or however many times a day that you eat, where you're driving muscle up and then it comes down and up and down, and voila, it's your mass that you carry with you every day. Now, one of the major ways to build muscle mass outside of just sheer feeding is through resistance exercise. So lifting weights or contracting your muscles against some sort of resistance. Okay, and so what happens is that resistance training can actually synergize with feeding to really boost the rates of muscle protein synthesis. So this is what this plot shows. So you can see here, if you're just feeding, okay, and what's shown here is kind of like, you're taking like a protein shake, for example. Okay, you'll get this sort of blip in muscle protein synthesis for, an hour or two after feeding. But if you've done some resistance training, okay, then you feed, you can see that there's a much greater increase in the muscle protein synthesis. Okay, and that this effect persists for many hours. All right, so let's say 24 to 48 hours. So what is the mechanistic basis of this synergy? Well, the way that muscle protein synthesis, the rate of muscle protein synthesis is really governed by mTOR signaling. Okay, and then a variety of other signaling pathways that kind of converge on mTOR. And so you can see here leucine. Leucine is the amino acid to which mTOR is the most sensitive. So leucine will actually trigger an increase in mTOR activity. And you also have insulin signaling, which will converge on mTOR signaling as well. And then here, glucose is shown because basically, it can also raise insulin levels, but this part isn't directly involved with the protein synthesis pathway. Okay, and one of the targets of mTOR is P70S6 kinase 1. Okay, and then this protein, you can see here in this figure, it's bound to EIF3, which is one of the eukaryotic initiation factors. Okay, so this is one of the proteins that will eventually form the complex that's called the pre-initiation complex. Okay, so by being bound to P70, that inhibits its activity. When P70 is phosphorylated, EIF3 comes off and is able to bind to the complex. P70S6 kinase also phosphorylates EIF4B, so another one of these initiation factors. Okay, P70S6 kinase also phosphorylates S6 ribosomal protein, which is one of the constituent proteins of the 40S ribosome. Okay, so it has other substrates. It's involved in many other different things, but for us, we're going to focus on it for its role in promoting translational initiation primarily. Okay, so when we were kind of getting into this area, the question is, okay, so we have this synergy. We could try to study, resistance training and nutrition together, but both have complex effects on their own. So what we decided to do is kind of study them separately. So we have one set of studies on leucine-mediated signaling and muscle protein metabolism, and then we have another set of studies on the mechanotransduction processes that cause the resistance training specific effects. So what I like to do is I like to develop mathematical models of the biological processes that we study, in particular kinetic models, because these models can really bring the system that you're trying to study to life and help you design better experiments, and you also can helps you quantify certain aspects of the system. So that's what we did here. So what I did was I tasked the PhD student, who's actually in the room, TJ over here, I said, listen, I want to understand when we take in leucine, which is like one of the major constituents of protein shapes and whatnot, when you take it in, what happens to it? Where does it go from mouth to muscle? So we designed this kinetic model, which is effectively, it's kind of like a pharmacokinetic, pharmacodynamic model, which you see a lot of in the drug industry, except for this is for leucine. So the initial part here represents leucine ingestion and digestion. And then the processes that occur in the blood. So leucine is absorbed and transported in the blood. It also stimulates insulin. So insulin's not just stimulated by glucose, it's also stimulated by amino acids, just not to the same extent. And then here we have a depiction of a muscle cell. Leucine can enter the cell, it can get incorporated into protein, or it can be oxidized. It can also help promote the activity of mTOR. Okay, so down below is mTOR. Here we have P70, and then through here we have insulin signaling, which is happening as well. Okay, so it's kind of I don't want to get too deep into this, but basically we have this kind of complete model. And what we did is we used it to study how the system is basically controlled. So we did some computer experiments like one would do in a lab. We overexpressed and we knocked down proteins like P70. Okay, and what we found is that when we overexpressed, quote unquote, P70, that it didn't affect how leucine was being transported or even AKT activity, but it did have a significant, substantial effect on muscle protein synthesis. So what I'm showing you here is P70S6 kinase. We have the total amounts here. We have 1, 2, and 4-fold increases in the total amounts. This is the phospho P70S6 kinase down below. And then what you can see here is 3 levels of muscle protein synthesis, which is the blue curves. And the solid blue curve is like the 1x or the baseline condition. Okay, and this is following a leucine bolus. And then when you overexpress P70, you get this dashed line. This is the 2-fold increase or overexpression. And then up here is with 4-fold increase in expression. You can see that you're basically you're effectively doubling the amount of leucine that's incorporated into muscle protein when you overexpress P70 by 4-fold. Now, we also went the other way, and this, I apologize, it's a pretty busy slide, but basically showing that the opposite holds true as well when you knock down P70S6 kinase. So these plots show P70S6 kinase levels. Here we have basically 1x, 0.5x, so we've knocked it down by 2-fold, 4-fold, and 8-fold. And if you just follow the blue line here, you can see how the solid blue line is falling in every condition. Okay, and then the corresponding total amount of protein, like the protein levels, these are shown by the green curves here, they're also falling. Okay, so effectively you're not incorporating, you're not building as much muscle with each meal when you have P70 knocked down. And you can't, what this figure shows, without going into too much detail, if you overexpress mTOR in response to try to substitute for that activity, it can't make up for the loss of P70 beyond a certain level. Okay, so these modeling results are like, wow, like P70, the absolute levels of P70 S6 kinase seem pretty important to muscle protein synthesis rates. And so what we did then was we looked into the literature, and we looked into the literature studying muscle protein synthesis in older adults, because what happens as we age is that our muscles shrink, okay, sadly. And so one of the reasons for this is something called anabolic resistance. Okay, so anabolic resistance is basically the dampening of muscle protein synthesis in response to anabolic stimuli like feeding and exercise. Okay, and so in this study, what they did is they fed older adults and young people with essentially amino acids, and they measured the levels of mTOR and P70 and some of the other proteins in muscle biopsies. Okay, and so what they found with the dosing of these essential amino acids, so with the feeding, they saw that the muscle protein synthesis rates were able to increase in the young up into a certain level, whereas in the elderly, those same amounts couldn't make as much protein as the young people, and even when they increased the dose to well beyond with the maximal dose that they saw for the young people, it still wasn't able to drive the system to incorporate more muscle. When they looked at the levels of mTOR and P70 in the muscle biopsies, the levels of these proteins are about half in the elderly compared to the young persons. Okay, so these are data, again, like we didn't use these data in building our model. We didn't really consider them. We didn't know about these results, or at least if we did, we kind of just knew about them in passing. But this was kind of corroborated than what our model was seeing. And our model was based on healthy adults. So some further studies that we did was to build a model or to change the model to reflect the mechanisms of anabolic resistance. So here we want to study now anabolic resistance using our model. And there's a bunch of different mechanisms that are thought to be occurring that cause anabolic resistance. So when we model each of these different mechanisms in isolation, and then we combine them, okay, you can see how this is normal, the normal spread of leucine incorporation into muscle, so the normal levels of muscle protein synthesis. Okay, and when you compare those to data, the gray dashed line is the rate of muscle protein synthesis in young persons, and this sort of burgundy line is the rate of muscle protein synthesis that was measured in older people. And these numbers here, these box plots, are all model simulations from models where we had different parameter values that kind of reflected like a population of individuals. Okay, and you can see that with these different mechanisms of anabolic resistance, the median muscle protein synthesis rate drops, okay, with some mechanisms causing worse drops than others. And when we combine them, you can see that that really drives the muscle protein synthesis rate really quite far down. But there's something called compensatory signaling. So somehow the baseline signaling for P70 goes up a bit, and that helps to compensate or offset some of these losses. And then you can see how the median of that simulated effect now overlaps the data from the older adults. Okay, and so then what we did is we studied how, if we go through and try to treat, create therapeutic strategies towards each of these mechanisms, can they recover the loss of muscle protein synthesis across all conditions? Okay, so here's where, like, let's say we had a failure, or we had an increase in splanchnic extraction. So that means like when you as the amino acids in your blood from your stomach go through the portal circulation, they get taken up by the internal organs. Okay, if that process happens faster as we age, that means you're going to have less amino acids to go to your muscle, for example. So if that rate is increased and we treat it, could it now can you then alleviate the effects of muscle protein synthesis? Okay, or if this is not a druggable target, if we target one of the other mechanisms and try to increase those rates, can it now compensate for the effect of the target that we can't drug? And the overall finding of this, and I'm not going to go through the details of the numbers, but basically by increasing the levels of P70S6 kinase in the muscle was the only mechanism that could compensate for all of these different mechanisms of anabolic resistance when compensatory signaling was present. Okay, and that's basically what this beige result shows. You're restoring it back to its normal, close to its normal levels. Okay, so all this to say, the absolute levels of P70S6 kinase seem important both in health and in disease for determining muscle protein synthesis. Okay, so if that's the case, then what determines the absolute levels of P70S6 kinase, or for that matter, any protein in our cells? Okay, and so what this slide shows is the sort of, the central dogma of molecular biology, but blown up a bit to include the mechanisms that we know about today in terms of how gene expression is regulated, and then protein translation and its post-translational modifications are all occurring to basically govern these levels. So at its most basic level, any given protein, its levels will be basically the balance between translation and degradation. But of course, there's processes that happen all around this. And in the case of P70S6 kinase, this is a map basically of all the regulatory post-translational modifications. Okay, and one of them, of course, is going to be ubiquitination. Okay, and a lot of the post-translational modifications are pretty well known in terms of phosphorylation sites, acetylation sites, and so on. But the ubiquitination sites are less well known. We know it happens within the kinase domain primarily, and it's mediated in part by a protein called ROCK1. Okay, so part of what's obviously, proteins get degraded. There are a variety of systems for degrading proteins. Ubiquitin-mediated proteasomal degradation is one of the major ones, and certainly this will help, degrade P70S6 kinase. Okay, so that has to sort of be true, but when we were kind of thinking about it, it's like, well, what governs the transcription? Okay, because that's like the first step in translation is you have to have the scaffold for to code for the protein. So that's what we kind of became interested in. And when I think about exercise and exercise training, we do a workout, and what happens is that exercise will stimulate a number of different intracellular signaling systems. Okay, and then I'll show you some details of which a bit later. And ultimately, these will converge on transcription factors that will then bind the DNA of fitness-promoting genes and either increase their expression or repress the expression of other genes. Okay, so in the case of P70, we don't really have a good idea of how this happens. We looked through different bioinformatic tools, just trying to get a handle on like what are the transcription factors that bind or that control P70 expression. And there's not a whole lot in the literature. And so then we decided to turn to bioinformatics and a set of tools under the family of causal network analysis. So these are tools that integrate omics data with structured prior knowledge from regulatory and interaction databases. Okay, and this is a recent review that looks pretty good. I haven't read through all of it, but this is a nice figure. And so this summarizes some of the contemporary tools that are available if you're interested in looking them up. But the one we settled on is something called Carnival, which is basically an acronym for Causal Reasoning Pipeline for Network Identification using Integer Value Programming. Okay, so I'm going to go through how this algorithm works in some detail, which I hope would be of interest to this audience. So there's a lot that goes into any of these different algorithms, and so it involves several databases and different tools. So we're gonna go through this workflow sort of line by line. All right, so we need to know what transcription factors might control the gene that codes for P70S6 kinase, which I'll just call the S6K gene for short. Here its official acronym is RPS6 KB1, so it's missing the B there. Okay, we then need what's something called a prior knowledge network. And this is like, what is this potential candidate signaling network that drives, that will ultimately regulate the transcription factors that bind to the, or that control the gene. We then need to know what transcription factors might be active in the context that we're interested in, okay, as well as the signaling pathways and so on. Okay, so we'll just go through this each step here. So the first tool that I'm going to tell you about is something called OmniPath. Has anyone heard of OmniPath? Okay, we got one. That's great. So OmniPath is basically a database of databases. So as we know, and you're probably well aware, like there's a gazillion bioinformatics databases out there. This one basically summarizes the data from over 100 of them. Okay, if you look carefully, you can probably find a database that you've used in your work somewhere in here. Okay, and so we used OmniPath for two distinct reasons. First of all, OmniPath curates a lot of the transcription factor regulons. So a transcription factor regulon is a transcription factor in the genes that it targets. Okay, and so some of these are predicted by data or shown by data. Others are predicted by like sequence motifs and things like this. The other thing we needed to do was to, again, get this prior knowledge network. And so what this network was in our case was we went into OmniPath and extracted out about 3,600 proteins, and they're interconnected by almost 10,000 edges or connections. Okay, so this is like a huge scaffold. We know that not all of these proteins or their connections are going to be either present or functional in our cells, in our cell type of interest and under the conditions of interest. But this tells us what this is the comprehensive set of possibilities. Okay, just to give you a quick vignette of how OmniPath can be used. Here we used it through the Cytoscape plugin. Okay, you just go into Cytoscape, you download the OmniPath plugin. It gives you a control panel. You can basically choose human, mouse, or rat. There's 4 different data sets you can choose from. Here I'm showing you the transcription factor target data set. Then it will include here, as your list of options, all of the different databases from which it's drawing from. You can select all of them or just a few of them, however many you want, and you can run the query. And then for your protein of interest, which in our case, or our gene of interest is SS kinase, it will give you, you can select out the nearest neighbors, which are the transcription factors that are thought to directly control that gene. And so when we did this, we got 19 different transcription factors from OmniPath. And what the colors represent is like where these transcription factors might bind, like either in the promoter, in the enhancer, or maybe in a mix of both. Okay, and the numbers are basically the total number of transcription factor binding sites in the enhancer region of the S6K gene. How many are within the promoter? How many are binding to the mixed region or to both? And then how many are binding to the enhancers? Okay, so there you go. Maybe you recognize some of these transcription factors. Maybe they're in your list of commonly expressed ones. Your list certainly may be reminiscent of this figure. Okay, so now we have our transcription factor regulons, and we have a scaffold, or the comprehensive set of signaling network that will be driving the activities of these regulons. So the next step is to estimate context-specific transcription factor activities. And for this, we're going to use a tool called Dorothea, which gives us transcription factor regulons thought to be acting when you give it an RNA or some sort of omic data set, a set of differentially expressed genes, in this case from RNA-seq. Okay, we're then going to estimate the activities of these regulons using an algorithm called Viper, which many of you may have used in here. Okay, and that will give us normalized enrichment scores, or NESs, for transcription factors. So the data set that we used is from 2018. It's a study in which the group studied aerobic and resistance exercise in 6 males who were recreationally trained. Okay, what they did is they delivered both aerobic and resistance exercise to the participants. Okay, and this is done in what we call a crossover design. So, in the initial visit, half of the participants did aerobic exercise first. They then had muscle biopsies taken at before exercise and then at 1 and 4 hours after exercise. So muscle biopsy is literally they're like sticking a needle into your leg and pulling out a little bolus of muscle. That's the really nice thing about studying muscle is it's one of the only tissues you can sample in humans ethically. Okay, so keep that in mind. And then what they did is they did a 1 week washout period. And then if they did the aerobic exercise first, they would do the resistance exercise second or vice versa. Okay, and similarly, the muscle biopsies were taken at the same time. If you look down below here, there's a bunch of numbers. It's basically showing the number of differentially expressed genes that either went up or down at the 1 hour and 4 hour time points in response to the aerobic or resistance exercise. And if you look carefully, and I know the numbers are small here and I apologize for that, but basically you're seeing more genes were differentially expressed in response to the resistance exercise, and most of the genes are being differentially expressed at 4 hours, only a few, or relatively fewer at 1 hour. Okay, which is kind of what you would expect, right? So normally when we look at exercise responsive transcriptional data, you typically need to wait a few hours before you really start to see a lot of the mRNA levels changing. Okay, so what is Dorothea? Dorothea is it's the acronym for discriminant regulon expression analysis. Okay, and VIPER is virtual inference of protein activity by enriched regulon analysis. Okay, and so again, this is to answer the question of the transcription factors that we predicted from OmniPath that would be responsible or could be responsible for controlling S6K gene expression. Okay, which ones are likely active in the setting of interest? In this case, this is in response to aerobic or resistance exercise in skeletal muscle. Okay, and then once you have the regulons, okay, or what Dorothea is, it extracts these regulons from multiple different types of data, like I kind of already discussed. Okay, and you can read the paper if you want to see the more specifics on that. And then you take these regulons and the differentially expressed genes, and VIPER will give you those normalized enrichment scores. So if we go through just to cut some example data here, here's 5 transcription factors in which they saw members of their regulons differentially expressed in the data set. This is in response to aerobic training. Okay, and so it's these results in red and blue that are really driving the selection of these transcription factors as the active ones. Okay, so this is for aerobic exercise, and then this is for resistance exercise. So there's one more example here. Okay, and so you can see that different genes are positively or negatively expressed, statistically significant, and over a 1.5 fold threshold. Now, for P70 itself, okay, so the mRNA for P70 S6 kinase, it was really only responsive in response to resistance exercise and variably responsive in the aerobic exercise. So that's the middle condition. So we have baseline at rest, and this is at 4 hours post for both of them. Okay, you can see that there's a clustering of points in the resistance exercise up high, and that its median value is quite high, whereas aerobic exercise, there wasn't really that much of a change. Okay, so when the data is run through Dorothea and VIPER, okay, these are the results we get for our 19 transcription factors that were thought to regulate S6 kinase. So you can see the normalized enrichment scores. The blue is for the aerobic exercise, red is for resistance exercise, and these scores can either be positive if you see an increase in activity or negative if you see a suppression in activity. Okay? Largely, you can see that both go up or down in response to either type of exercise, but not exclusively so. All right, and that some have higher scores than others. Certainly, genes like HIF1 alpha, that's like a major known exercise responsive transcription factor. So that makes a lot of sense that it's there. So that kind of validates in part that what we're seeing, that the algorithm is at least pulling out effects that we would expect to see. Okay, so that's the TFs and the transcription factors and their activities. Another input to Carnival is something called like the signaling activities. And the way that this is estimated is through an algorithm called progeny, or pathway responsive genes. This is a bit complicated, but basically all it is the data for this came from something called perturbation experiments. So suppose you have a group of cells and you add to it a cytokine that you know elicits a certain signaling pathway, such as EGF, the epidermal growth factor. It binds the epidermal growth factor receptor. It will stimulate MAPK signaling and some other signaling pathways. And there's be a sort of a standard set of genes that will probably go up or down in response to this treatment. Now imagine you do hundreds of these types of experiments with different compounds targeting different signaling pathways, and you measured all of the differential gene expression, okay, such that you could then develop a matrix where you have sort of a set of genes that will consistently reflect whether a certain signaling pathway was activated or not. All right, and then if you have that kind of matrix, what you can then do is you can feed the algorithm with the set of differentially expressed genes, and it will sort of back estimate what signaling pathways were likely activated in response to that perturbation. Okay. So we ran this algorithm as well, again, using the Dickinson data. Okay, and there's these set of signaling system. You can see there's a normalized enrichment score. And then in general, the signaling tended to either go up or down in response to exercise, irrespective of whether it was aerobic or resistance. Some signaling pathways maybe a little bit more for the resistance training. And the reason for that is probably the resistance exercise that was done was 8 sets of, it was a pretty heavy weight. It was a pretty severe stimulus by comparison to the aerobic exercise. So this result here is not entirely surprising, given that there's a bit of a relative difference in the severity of the two stimuli. Okay, so that's, now we've kind of filled out our most of our inputs. There's one more input that we can feed into Carnival. Okay, and before I kind of get into that, I should note that you can run the Carnival algorithm in 2 modes, standard and inverse. In the standard mode, this is where the user provides targets of perturbation. Okay, so this would be, you're giving it, the genes that you, or in fact, the proteins in this case, that you know will be responsive to exercise. Okay, or you can let Carnival run without giving it targets of perturbation and let Carnival infer what those targets are itself, just based on the data. Now, in exercise, there's several canonical signaling pathways that we know get activated in the muscle. So calcium signaling is a huge one because when you have an action potential and a contraction, it requires calcium signaling. Calcium will then bind to calmodulin, and calmodulin will activate CAMK2 signaling, so calcium calmodulin dependent protein kinase, as well as calcineurin. And both of these signaling systems have pretty significant impact on the muscle fiber type and different properties. AMP activated protein kinase is another major signaling system that gets activated with exercise. When you exercise, you're obviously using a lot of metabolic energy. You're breaking down ATP into ADP and AMP. And this protein directly senses the levels of those ATP breakdown products. Okay, and then it gets activated, and it basically tries to stimulate energy production in the cell. Hypoxia-inducible factor. Again, when you exercise, you drop the intracellular oxygen levels like almost tenfold, okay? Like quite a significant amount, enough to elicit HIF1, the accumulation of HIF1 alpha, and then they'll bind to HIF1 beta and then elicit HIF1 signaling. And then mechanotransducers like focal adhesion kinase will respond to the force of contraction. And then that mechanotransduction and reactive oxygen species will tend to stimulate the MAPK signaling pathway, which again tends to get activated under many different conditions. Okay, so we know all these things are going to be activated in this study and that are going to be driving a lot of those changes in those gene expressions. So what we did is we fed to Carnival 50 targets of perturbation based on these canonical signaling pathways. So, again, the details aren't super critical here, but we basically fed it in the genes associated with ADK, calcium, hormonal signaling, so protein kinase A, and the NAPKs, and so on. Okay, so now Carnival has all the inputs. We have all the inputs prepared. Now what we have to do is to run the algorithm. So what Carnival does, it uses integer linear programming, the CPLEX algorithm in particular, okay, to minimize a function. And this function seeks to generate networks, okay, that best explain the node activities inferred from the data while minimizing the number of nodes, total nodes in the network. Okay, I'm not going to go through all the details of this equation, but this is sort of it. There's two parameters, alpha and beta. One's associated with something called the transcription factor specific mismatch penalty, and the other is a node penalty. And I believe it's beta that you can manually tune, and alpha, I think, is based mainly on the Dorothea outputs. Okay, and then this other number here, gamma, that's where the progeny data enter. So all of those inputs are entered, find a way into this equation, and then Carnival's trying to minimize a function that best, to give you networks that best explain the data. Okay. All right. So first of all, I'll show you some runs where we fit networks using the standard Carnival algorithm. Okay, and I apologize again, like I'm going to show you these networks. It's probably not very easy for you to see, especially in the back, but I'll just sort of try to take you through this as best I can. So what the networks look like is you have three different shapes. One's a hexagon. This is our targets of perturbation. So we told Carnival that these have to be that these are active. Okay, so it knows that. And then anything that's a sort of an oval is a node that was inferred to be present in the networks. And then anything that's a diamond is the inputted transcription factor. It's one of those 19 transcription factors that we put into the algorithm. Okay, and so what Carnival is doing is it's basically telling us what nodes and then what connections. So you have these edges. The edges are arrows, or like the sort of T-ended arrow, which signifies inhibition. So the regular arrows are stimulation, the T arrows are inhibition. And you see that there's different colors and different thicknesses and darknesses of the lines. So what you do when you fit Carnival networks is you don't just fit one network, you get many that sort of equally best fit the data. And then you can aggregate those into a single consensus network. And when you do that, anything that's colored blue is within that consensus network. Most of the activities for that protein were negative. Okay, so they were reduced. If they're red, that means the vast majority were positive. If they're closer to white, that means some of them were negative, some of them were positive. Okay, the edges work kind of a similar way. If you have a black and a thick edge, that means it was prevalent in most of the networks that were fitted. If it's lighter in color and thinner, then it was only present in a few of the networks. Okay, and so what we can see here is we can see this HIF1 alpha. Okay, it was positive, right? So like its activity was increased, and you can see that all of the things feeding into it are known. It's known hydroxylases, so the prolyl hydroxylase, the PHD enzymes. Okay, so this is sort of validates what we know to be happening, especially in aerobic exercise. We have AMPK over here feeding into a transcription factor called SNAIL1. And SNAIL1 is actually a repressor. So by reducing its activity, we'd actually be alleviating repression on some of the target genes. Okay, we can go through all of these networks in a similar way and try to pick out what the proteins are and what they do. And what I'll do is I'll show you the networks and just come up with some summary observations. So this is the network for resistance exercise. It's got more components than the aerobic exercise. Again, resistance exercise seemed to be a bit more of a profound stimulus for the muscle in this study, and that may underlie why you see a bit more of an extensive network. Okay, so now I'm going to show you the two sets of networks for the inverse, from the inverse Carnival. So they will look different because here what we have is at the top we have the perturbation, which is exercise. And then these are the targets of perturbation that Carnival guessed must have been activated by exercise. Okay, and you can see they're all pretty faintly colored. meaning that we didn't get like a really good set of target, a set of certain targets of perturbation from this algorithm. Okay, the resistance, and this is just blowing up what those targets of perturbation are. You can look carefully to see if your favorite transcription factor or signaling protein is in there. Okay, you can see that of them, most of them are pretty white. Okay, except for the last two at the end are slightly red, meaning that we have in most of the networks, those two transcription factors in this case were activated. For resistance training, we get something similar. Again, our targets of perturbation are mostly colored white. We have two on the end here that are colored darker blue, meaning for the most parts their activities were suppressed. Okay, and I'll just blow those up for you, just so you can see. Okay, but the question, really is, so what are we going to learn from these networks? So you have to kind of now spend time staring at the networks and like going through and figuring out what proteins are coded for by each of these genes. So I'll just highlight a few observations for you. I've already mentioned that HIF1 alpha was increased in response to both types of exercise. We would expect this. We also saw that the SNAIL1 protein was suppressed in response to both types of exercise. So that's just kind of interesting. There's no real literature on SNAIL1 and exercise responsiveness. So this would be interesting, something to just follow up on if we're interested in exercise responsive signaling. There's also this protein, gene ESR1, which codes for the estrogen receptor alpha. And estrogen receptor alpha is a known transcription factor that controls S6K gene expression. Okay, so that's really interesting. And we know in response to exercise that estrogen levels go up, even in males. Okay, so males do have estrogen, they're just at lower levels. Okay, so that may be why we're seeing a bit of an increase here, and in particular in response to resistance exercise. One of the other results that would have been very hard to see, these are just sort of cutouts from each of the networks, is that every network that we fit featured suppression of phosphatase activity. Okay, as one of the top level nodes as well, okay, which is pretty interesting. Like normally when we think about exercise, we think about it activating a bunch of different things. We don't normally think about it suppressing the suppressors. And so this has received relatively little attention, but maybe that can, there's a general program where you do get a suppression of inhibition to allow for the ways of gene expression and so on that follow from exercise. Okay, but we still haven't answered the question that we kind of set out to address at the start, and that is, how is S6K gene transcription controlled? And remember, I showed you these data. Okay, we only really see its mRNA levels go up in response to resistance exercise. So a lot of the results that we saw, in terms of the transcription factors being activated in both cases, don't explain that result. Okay, but the ones that do, we do see some differential activities of these transcription factors that I've highlighted here in turquoise. Okay, so if you can go through, there's three here where we see an increase in resistance exercise compared to the activity we saw in response to aerobic exercise. We also see this transcription factor here, DLX4, where there's a marked suppression by aerobic exercise and not much change in response to resistance exercise. Okay, we can continue and go through and see differential levels. Even in this case here of FOXA1, we see that it's suppressed in response to aerobic exercise and not really changed in response to resistance exercise. So there's different modes by which you get the sort of differential effects of the two exercise types. Okay, so this is kind of interesting. There's certainly food for some hypotheses that could be then followed up experimentally from these results. Okay, so there's a number of limitations, as you can expect from these types of methods. Here we're using an RNA-seq data set from just 6 participants, and they're all males. Okay, so that doesn't help us in terms of learning about female physiology, which is relatively understudied compared to males, and that there's a major push in our field to address. We're also I didn't really point it out, but NF-kappa B was the top, it was the transcription factor that had the highest, or the signaling system that had the highest activity in the progeny results. When you do exercise for the first time, or if you're doing a mode of exercise with which you're unfamiliar, you'll get a muscle damage response. So most of you have probably felt sore in response to exercise, and especially when you go to the gym after you haven't been at the gym for a while, or you go for a run, okay, because that's high impact activity. You're causing eccentric contractions. That is contracting the muscle while it's lengthening. Okay, and that is the major type of contraction that causes muscle soreness. All right, so if you're doing unaccustomed exercise, you're likely going to see an inflammatory response in the muscle because you've caused damage to the cells. The immune system is infiltrating, and there's an actual inflammatory response. Okay, also, we're studying here, we have a general question about S6K gene expression, and we've only studied it here in response to exercise. Okay, to actually get a good handle on it, we'd have to take a bunch of conditions like from GEMMA. That looks like a really great tool. So where we could take, human data sets from all sorts of conditions and then see what transcription factors might be activated across all those different conditions. The inferred networks are inevitably going to be biased towards known biology. Okay, so all the databases are effectively known biology. It can certainly help to identify new connections, but you're not going to find necessarily new molecules or anything using this approach. And then just the interpretability issue. When you get presented with a network, okay, with these different colors and the different edges, and it's just very complicated to look at, and you're like, well, I don't even know what to do with this, right? It takes a lot of time to kind of go through it. We've gone through a number of the different transcription factors and tried to pull literature to try to learn, does this make sense? But ultimately, that's a rationalization, right? We're trying to rationalize our results, and to validate any of this, you have to actually, come up with predictions and then validate experimentally. Okay, and then finally, obviously, this has just sort of helped us to infer the transcription, the regulation of transcription of the S6K gene. But really, its protein levels will ultimately be governed at all sorts of levels of regulation. So that's just one of them, and we haven't really, looked at the others. Okay, so just to summarize, hopefully I've shown you how the absolute levels of P70S6 kinase are important for determining the muscle protein synthetic response and ultimately then your muscle mass. Okay, P70S6 kinase is controlled at multiple levels, and its dynamic transcriptional control is poorly understood. We used one of these causal network analysis tools, in this case Carnival, to infer what that network might be in response to exercise. Okay, I showed you what the inputs were and what the different databases and algorithms that went into it. Okay, and we've gained a few insights into exercise responsive signaling and some hypotheses about how S6K transcription might be occurring in response to different types of exercise. So with that, I will thank you very much for your attention. All this work was done by TJ McCall, who's here, and he's now postdocing at BC Cancer, just down the street here, looking into PK/PD modeling for personalized radiopharmaceuticals. He was helped a lot by an undergraduate researcher, Alex Zhang, who now I believe is applying to graduate schools. And then we collaborated very closely with Aurelien Gourd and Julio Saez Rodriguez at the European Bioinformatics Institute and the University of Heidelberg. We really depended on them to help us get us started with their algorithms. And then TJ's PhD committee was made up of Eldon Emberley from SFU, Dan Moore and David Church from the University of Toronto and the University of Arkansas, respectively. And this work was funded by INSERT. So thank you very much again. Yes.
Yeah, thank you for that talk. It's really interesting. I just had a question for the diagrams that you're showing. The different pathways and stuff.
Mm-hmm.
That you had.
Yeah.
So I was just wondering, you're getting sort of things is driven by the fact that there's maybe a lack of consensus between the actual binding data for the transcription factor and the expression data?
It's a great question. So I think the main reason I think for the wider color, you can see here that there's redundancy, right, in several. So Carnival can't decide, if there's isoforms that are converging or several signaling pathways that are converging on a target, and any of them are equally plausible, then you can imagine how we would have a very tough time deciding, is this positive or negative? They'd be offsetting each other. That's what I think is happening in most of the cases. In terms of I'm not an expert at all on the quality of the transcription factor binding databases. Again, Dorothea would be pulling evidence from all different types, like actual ChIP-seq experiments, but also transcription factor binding predictions and so on. And I suppose that could, there could be some conflicts in there. I'd have to really go through the OmniPath papers in detail to see what kind of quality control that they did. These guys are rigorous, at least as much as one can be. But when you start to deal with hundreds of thousands of data points, then you can imagine that there'd be some breakdown. And then also conflicts between cell types, because OmniPath takes in data from every cell type, and of course, you're not going to context specific signaling is exactly that. You're not going to have everything operating in a given cell at once. There will be certain pathways emphasized versus others.
Thank you.
Yes.
Hi. Does this work?
You have to press it.
Okay. Oh, it's okay. I can hear it. Great talk. Thank you. I'm wondering about convergence, because you mentioned that you believe that there are many transcription factors which control the expression of the kinase, and that they all converge on regulation of that kinase. But who says that?
Okay, so I'll just, if I understand your, are you kind of raising the question, do we expect all 19 to be on the enhancer at one time driving the expression? Is that what you're kind of asking me, or?
No, I'm asking why you believe there's a convergent pathway that regulates this one transcription factor.
Okay, so
that is responsive to exercise.
Okay, so when you say convergent, there's a bunch of pathways converging on P70?
Well, that's
kind of what I'm contending, yeah.
That's the slide, yeah.
Yeah, okay.
There's a slide on that.
So, well, I mean, I guess that's the hypothesis, right? I'm not saying that that's necessarily true, but the fact is, in most cases, you can't take a single gene or single pathway approach and explain the variance in transcription, for a given product under normal circumstances, right? So, I mean, kind of going back to the days of when systems biology came around, and we kind of had a very distinct pathway, one gene, one pathway, one function type of assumption, right? So hypoxia is an example. Hypoxia, you activate hypoxia inducible factor, and then that's going to increase the expression of genes that deal with hypoxia. But I think there's been discoveries where now it's operating under conditions which may not necessarily be in response to hypoxia, and it may affect certain genes in that condition and certain genes in others. We also know that these pathways are promiscuous. There's crosstalk. So, I mean, it's been a long time since I've read one of these specific review articles on the basic premise of systems biology, but what I've seen in my own data, I think there's stereotyped pathways. If you apply hypoxia, HIF is going to be the most active pathway by far, but you'll see some others maybe coming on or off, and if you mess with those might change your output to some degree. So, yeah, I guess my worldview of biology is that the signaling system is a network that multiple, and the other thing too is the hourglass effect, right? There's relatively few signaling pathways that then have thousands, hundreds of thousands of stimuli and thousands of genes that they have to regulate. So if you only have a limited number, eventually you have to have some this convergence, or at least a crosstalk, where a relatively small network is driving a lot of changes. Does that somewhat answer your question?
Yes, thank you.
Does it convince you that there's convergence, or we'll have to talk at dinner? Or
We'll have to chat about it.
I look forward to being convinced otherwise. Other questions? Yep.
Yeah, I was curious here. So it sounds like it would be great if we can find some way to increase P70S6 kinase so we can get stronger or grow more meat or what have you.
Yeah.
Are there any findings so far that look like a promising way to get to that more, applied intervention?
Not that I'm aware of.
Sure.
That said, I mean, there's plenty of research on trying to artificially increase muscle mass, but also create exercise mimetic drugs, for example. So I'm much more familiar with that. So I can tell you a little bit of the stories about that. But there are definitely pharmaceutical companies that are testing compounds that activate AMPK and usually something like PPAR transcription factors, where at least in animals they can increase running endurance. So like in mice, if you treat them with this, they can run further on a running wheel. You can also treat. It can be helpful for those with chronic conditions or obesity who would struggle to burn enough calories to promote fitness because they're that unfit or that weak. That's where you'll start to see exercise mimetic drugs coming in and intervening. So whether it's something similar just for increasing muscle mass, I mean, obviously people injected anabolic steroids forever and other pretty crude methods with very significant health side effects. But that's sort of that's the story. Okay, so you can intervene on these things, but if you're using some sort of crude drug, then you're going to have pretty potentially nasty side effects. But yeah, I mean, I think that this type of research, and there's plenty of research ongoing that is getting closer to that reality.
Yeah, that makes sense. Thanks.
Yep.
Builder side of things, and we don't work exactly on quite the tools you're using.
Okay. Yeah. So, I mean, TJ would be the one to ask about the frustrations. We definitely had some. All I can say, for the carnival part of it, they did it was a huge, but there's a pretty extensive and complete literature where they've compared carnival and benchmarked it against other algorithms, such as causal R is one of them in the original carnival paper that they compared against. There's now in the review article I was just mentioning, and also I'm familiar with a kind of a related algorithm program called signaling profiler. I've never used it. I don't know much about it, but it's kind of a newer offshoot of Carnival. So yeah, no doubt that there's competitors, probably each with their advantages and limitations. I'm not super familiar on should we have used a different one? I think it would have been a trade-off, different benefits, different problems, because that's kind of where we're at, I think, in the field. But I think the exercise, I was asking TJ, is this all worth it? I have no doubt that it was worth it for us in terms of learning, how to use omic data and actually getting, a more significant hypothesis other than just going into regular pathway and network analysis methods, which are really, to me, very unuseful, right? Just the basics, these vague pathway terms, it just, for me, does nothing for me, inflammation, oh, great, thanks. Protein synthesis, great, oh, who would have known? Yep.
I have maybe a more specific question that's building on kind of the utility side. For a figure like this, you have kind of a spectrum from blue to red, but you probably lose information about where that, positive and negative effects are coming from. Would you have found it more useful? This is a very niche question, I'm sorry. But instead of having an overall color, maybe to have some distribution of what is blue and what is red, so you actually see where proportions are coming from, instead of just the overall aggregated.
Yeah, it's a great idea. I think what can I say? Yeah, just show like, yeah, the distribution would be an interesting addition to the visualization. I mean, like what I would really like is in a consensus network is for the colors to actually represent a quantitative measure of activity. That's ultimately what I would really like, because again, to answer the question, what is R6, what is S6K gene, likeliest, what are the driving factors? Then you need, some measure of how active are they, right? In a more tangible sense. So that's actually what I really like. But I think your suggestion for building in some context also makes sense.
I can ask another question.
By all means.
Oh. Okay. So since Thomas asked about like therapeutic target development, I wanted to ask, since this signaling pathway and the gene is implicated in a lot of cancers, like, do you think that's cause for concern? I mean, it's just seems like a generic cell proliferation signal, or
yeah, you're talking like S6 kinase being important for a lot of different things.
Yeah, well, it seems to be important for cell proliferation, and it seems to be involved in like certain cancers or just like general tumor development.
Yeah, I mean, I think that's always the concern, right? If you intervene specifically on any given protein, that it could you get off-target and side effects that could be deleterious for sure. So the other thing to consider here, like our model, our endpoint was P70, but like there's a whole set of proteins that are responsible for translation, and the quantities of those proteins are ultimately going to matter, not just P70, right? So I don't know if like our model might be showing that it's the protein that's really important, but it may just be that the overall amount of translational machinery is important, which would make sense. And that's one thing that happens in exercise. There's something called like there's a not only an increase in the sort of the initiation and so on, but like also just the amount of machinery, like the amount of ribosomes and such goes up with training. So you kind of like hit it from both angles, like the stimulus for increased translation, but also like the capacity to increase translation. So I think the story will go beyond, in reality, will go beyond P70. But I still think like we, as a field, never really focus on absolute protein quantities. And at least like, I don't know, even before this in my PhD research was on something completely unrelated. Like I made the effort to quantify absolute numbers of a protein in a cell, and I have no regrets about doing that. I think that's a really good thing to do. And like, we have mass spec-based methods now that you can do absolute quantification. We need to start really looking at stoichiometries and one of the ideas that we discussed is like just going through and using some of those proteomic data to like put numbers of how much of these of the corresponding protein is present in the cell, right? Because if you have 10 copies of this and 100 of this, well, your pathway, your information flow is likely going to be biased towards the side with the highest numbers, in theory.
Thank you.
Okay, thanks.